Hey all, in this video we are going to talk about metagenomic data quality control tools.
Exactly is metagenomic data? Metagenomic data refers to the collective genetic material recovered directly from the environmental samples, representing the genome of a community of organisms, often microorganisms within a particular habitat. Unlike the traditional genomics, which focuses on individual species, metagenomics analyzes the entire collection of DNA in a sample, allowing scientists to study the diversity, structure, and the function of microbial communities without needing to culture them in the lab. Metagenomic data typically result from high throughput sequencing or the genome sequencing, which means the rapidly produce vast amount of DNA RNA sequence data in a single run, and which also provides information of the composition and the potential microbial communities across various environments, including soil, water, and the human gut. And in this video, we are going to talk about the data that we've been collecting from a shotgun metagenomic sequencing. So in the case of shotgun metagenomics, the data contains random DNA fragments from the whole community, which can be assembled and analyzed to identify species, genes, and metabolic pathways, and also functional potentials. This flowchart here describes the main step in the metagenomic analysis, starting from the raw sequencing data typically obtained from the microbial communities, and then moving on to pre-processing and quality control that ensures the data is clean and ready for analysis by removing low quality reads or any kind of contaminants. And then moving on to the alignment-based analysis or assembly-based analysis. Alignment-based analysis typically means that reads directly to reference databases and which allows for quick annotation and taxonomic identification, and assembly-based analysis that reads into longer contigs or continuous sequences, which helps in reconstructing genomes and identifying functional genes. And this goes to annotation. There can be taxonomic annotation and the functional annotation. Taxonomic annotation typically means that identifies the species or taxa present in the genomic sequencing, and functional annotation refers as the assigns biological function to genes or pathways within the microbial communities. And this goes to the downstream analysis, which is divided into the diversity, phylogeny, and taxonomy composition. And the last step is the data analysis and the visualization, by which the integrates and visualizes all results, providing insights into the community composition, diversity, and functional potential. So today's main approach from this video would be focusing on the raw data and the pre-processing and cleaning of the raw data, which is the quality control, which is really important because with cleaning or pre-processing, it's really essential to do because it makes the data more accurate, reliable, and ready for meaningful analysis. Because otherwise, the data may contain very low quality reads or adapters that are a very high data complexity that's gonna be make the data more complex when if it's going to analyze, or it can be giving very low quality data results, which is not expected from the analysis. So data cleaning and quality control is really important.
When working with sequencing data, ensuring the accuracy and quality of your data is the critical first step. This slide summarizes some of the most commonly used tools for data quality control across different sequencing technologies, whether it's short read, long read, or linked read. So, FASTQC is most widely used to generate detailed visualization report of raw data set, and is often paired with tools like Trimmomatic to clean up any flagged issues. And there's another tool called Multiqc, which is a powerful tool that simplifies the process of working with multiple datasets by aggregating the results from quality control tools like FASTQC, Trimmomatic, and many others. So instead of manually reviewing separate reports, MultiQC combines them into a single file to navigate into. And this feature is particularly is useful for large projects involving multiple samples. For example, if you're running FastQC on dozens or even hundreds of sequencing files, you can use MultiQC to create a summary that highlights key trends and quality metrics across all of your samples. This is not only save you time, but help you spot outliers or systematic issues in your data. So for today's tutorial, I will focus on first QC and demonstrating how to run it and interpret its visualization. Afterward, you can consider using MultiQC to compile this output if you are working with a large data set. I've included links to all of the tools mentioned in this slide in the description below, so you can explore them based on your project needs. So let's dive into the demo and see how to use FastQC for sequencing quality analysis.
So once you have your raw data from somewhere, if you have this on your hard drive, then it's fine. If you don't have it, if you have the link, you can download it by using the wget command. So first thing to do is to log in into your supercomputer. You can do it from any remote system like PowerShell or ThinLink. Here I'm using ThinLink, but you can use any other remote server as well. So at first I will be logging into my supercomputer account. So that's what I'm gonna do now. So see.do. Then it will ask me about my password. Then I'm gonna just print my password, and then it would ask for authentication. You can use any kind of authentication that you want to, but I prefer to use the duo push. Now it's done. I will check which directory I'm in by using pwd. So it says I am at my home directory. So I will check the list. So you can see the list in here. So this is where I have put my raw data, but if you don't have it and you need to download it, you can use wget and the link, and use a period. If you are already in the directory, if you need to make the directory, you can make the directory by using mkdir and the name of the directory. Then you can download the uh, all your data in that particular directory. As I already have all this data in those directory, I am just gonna change my directory to that data. So now I am in the directory where all my raw data are in now. I will check what's the list of the directory, and it says, okay, so these are the my samples and or my sequencing data that is around the directory. So first thing I need to do is to run the first QC of my sequencing sample data. So I already have prepared my first QC um, script. I'm just gonna open it. So using nano, and here is my first QC script. So you see, this is the first shebang line. This is I have put it. I need to like download it. The wget source on the link. And so first thing about to keep in mind is that before running the script, you need to load the first QC module, like based on the uh, like the latest um, based on the latest, I guess, the module that's currently running on. So you can load the latest first QC version. You can also check the first QC version if you have any. If you don't have that, then you can just load the any version that you want to load, and then you can set up a loop for the first QC file. And I have used this wildcards asterisk because all the sample file that is my raw data sequencing file has the same extension, and using this asterisk would indicate all the files in the same extension. And by creating this loop, it's gonna run the first QC for each file for in each iteration. And if output is done, like once it's done with iterating all files, it's gonna stop at this point. So if you want, you can also put a printing statement is there, like okay, if it's done or not, if the file exists or not, something like that. So I'll be exiting my command line now, and I will just give to my command line. So once I'm in my command line, now I have set up my script. Now I can run the script. So for running the script, I'll put dot / and my script, and then I can press enter and run the script. As I'm already in the directory, I don't need to put a file because it's gonna already read the files it has in here with this file extension, that is fq.zz. So as I've already run it already, because it takes some time to run it, so and I have got this output file called FASTQC.html that I need to read the output. So as I have it now already in the supercomputer, I would need this file to save or copy in my remote computer. So for that, I would need to open a new tab, and it will just show that which directory I'm on. So I'm in my own home directory. So I would change the directory now to the directory that I have created to save the output file. So as if you haven't already created a output file directory, you can create it using the mkdir. A few things that I haven't mentioned earlier in my first QC scripting. So for make a script, you first need to, so for directory you use mkdir, right? But for making a new file or any kind of like not a directory file, you can use the word command touch and type the script name or file that you want to make. So for us it's like fasta .sh sorry .sh. In that way you can make your FASTA script file, and you can execute it using chmod plus x, then the name of your FASTA sh, or any file name or any script name that you have given it into, and in that way you can execute your command file, or a script file. And if you need to open that, you can use nano. Nano is kind of a text editor you can use. So using nano you can open and edit your script. Alright, now that I'm in the directory that I want to save my file into or copy my file into, I would just need to put the command secure copy, that is a cp, and then I would need the path of the file, like the source where it is right now, that is this one. Okay, so I have copied this, and then I will go to the here. So here at first I would need to put the okay, oh, I don't know why it's not showing me, but I have done it just a few minutes earlier. So secure copy, then you would need to write your supercomputer account, user 1 22, then you would put a colon, then your path. So if you put the period after it, that means it's gonna read that you're commanding it to copy that in that directory. So if you run it, it's just gonna copy the file in this directory. And so mm, as I have already done it, so I will just show you the file of the HTML file that is there. So you see the HTML file is already in here, the one that you're talking about. So now we're just going to open and see how it looks like. Let's go to the microbiome directory from my home directory, and I hope this is the one. Okay, so here it is. So now you can open and see summary of your first QC report. So it shows you like some basic statistics like how much is the content, your base quality, um, or base sequence quality, your per tile sequence quality, and this is the sequence quality scores that you can look into. It's like all kind of it's, and this is the per base sequence content, like how much of percentage of TCAG you have. You see my data has some kind of noise in here that I would like to get removed if I want to have a clean and a good quality data. My GC content tells me it's pretty much okay, so it's not that much of a concern. And my adapter content also says okay, so it's the green tick means okay, it's fine. This yellow, it means that okay, there's some concern that you need to fix or you can look into. So that's how you can run FASTQC in from your data files, and you can get the first QC report from your file. That's how you can get the quality report from for your sequence data.
So these are the two references that I have used for this video. So if you want to know more about the contents that I've used, you can go to this papers. You can find it in Google Scholar or PubMed. Thank you so much for watching my video. Hope you would find it helpful working on the quality control of your sequencing data. If you have any question, feel free to comment down here. Thank you.